Okay, guys, to go along with that quaternary structure we were just talking about in the last video, I just wanted to show you some examples really quick. You can see that, here we have three different types of protein, right? This is the three-dimensional structure of the proteins, uh, you know, uh, well, different proteins in question, right? You can see that this one is kind of globular, right? So they call this globular. Looks like a little glob, right? And as you can see here and here, you have chains of polypeptides, which are pretty much oriented, like a rope. Collagen is a real, very linear protein, so it's kind of like a string. And then you have this more, again, globular protein over here, which is hemoglobin. So hemoglobin is quaternary because it has two beta chains and two alpha chains, and they work together to hold well, oxygen for your blood, which we'll eventually get into. But um, this is really just a three different examples of how different proteins, in other words, different polypeptides, can work together to form a new structure, right? So how I generally think about this, um, between primary, secondary, tertiary, and quaternary, I usually try to tell my students, think of a roller coaster. I don't mean like the big wooden ones. I mean the really nice, smooth, fast metal ones, right? Well, if you think of a length of track, right, that would be like the primary sort of structure of a protein. If you think about like the one loop that's going into it, or maybe two loops, right, that would be like the secondary structure, primary over here, secondary. If you think of the entire roller coaster with all the loops and twirls and twists and all that other stuff, right? That's tertiary. That would be the whole roller coaster. Okay. Now, I don't know how you do this, but if you jammed a couple of roller coasters together, that would be like the quaternary structure. Okay, so generally that's usually pretty helpful, where you go from the single track, right? That would just be a little tiny piece, or just the sequence of how the roller coaster was constructed. That would be primary, and then you start to get into smaller and smaller shapes, and once you put all those shapes together, you're in tertiary. More than one roller coaster put together would be quaternary. Okay, so what you're looking at here are different three-dimensional shapes of the same protein, right? One is considered a ribbon model, right? And one is considered a space-filling model. All of these little colored dots in here are individual atoms in this protein molecule, right? This represents more of like the shape, like the structural functional shape of the protein, right? So I just want to, bring this up. It's not anything you ever have to memorize, but I want to make sure you understand that the three-dimensional shape of any molecule, including proteins, is really what allows it to function in a living system. If you change the shape, you change the function. Okay, so we already talked about what can alter or, lead to that functional shape of a protein. It's the number of amino acids you have, the number of monomers, in other words, and also the sequence of those monomers, of those amino acids, right? So that's going to determine the final protein shape, okay? Keep in mind, though, the result of anything, can be altered by the environment, right? So, if you're talking about a protein floating around in the human body, if the pH changes greatly or the temperature changes greatly, that can alter the shape of the protein, even though the sequence and the number would be the same. Okay, so let me just talk about pH first. Um, my example for this one is if you've ever eaten Sour Patch Kids or like those really super sour candies, right? And say you take that Sour Patch Kid and you just suck all that super sour stuff off them on the same part in your tongue all the time, and I bet you half of the class has done this before at least. You start to get that little like numb spot, on your tongue, and it kind of feels like, I don't know, a little bit different. What's going on there is that the amount of acid in the candy, the citric acid that they put in the outside of the candy, actually will start to change the shape of the proteins that make up your tongue, which is really kind of unexpected, I guess. But that's why your tongue feels so weird, after you eat enough Sour Patch Kids. So we call that change in protein shape denaturation. So in other words, you're going from the natural shape of the protein, which would normally be on your tongue, and you're changing that shape by putting it in an acidic environment, right? By putting too much acid around it, which is a change in pH. Okay. This can also happen with salt concentration, but what I really want to focus in on is temperature. What I'll actually ask you out of these four different factors here are about pH and temperature. Temperature, if you know what makes a hot substance hot and a cold substance cold, it's molecular motion, right? So in other words, the faster the atoms or molecules are moving in an object, the warmer it actually is. Okay, so if you think about it, if you put protein in a pan, and put it on the stove or in the oven and you cook it, what that temperature is going to do is change the shape of the proteins again. So my example for this one is, you take a raw chicken breast out of the freezer, right? So you're going to cook something, chickeny, right? If I was asked you to describe that chicken breast, you could say, well, it's kind of floppy and, it's kind of translucent. You can kind of see through it on the thin parts of it anyway, right? Okay, so take that chicken breast, which was cut right off of the chicken's breast, of course, right? It's the pectoral muscle of a chicken. You put it in a pan under enough heat, and what happens? It starts to change color and it starts to change texture, right? It's no longer floppy anymore. It's kind of, well, it's more stiff, right? You can't really see through the edges anymore. They're more opaque. Well, what happened there was the proteins moved so fast, in the chicken breast, they vibrated so fast with the heat that you applied that they changed shape. You can't take a cooked chicken breast and make it raw again, right? So it was a chemical change for sure, right? So, this also happens when you burn yourself. I mean, it's really the same thing. You burn yourself, you touch a hot iron or stove or, whatever you burn yourself on, right? When you were cooking. And, you get that blister, that change in the sort of like the color of your skin briefly, right? Well, what you did was you denatured your own proteins. You cooked them and you can't use them again. So your body has to get rid of them, heal, and then eventually build new proteins that are back in that natural shape. Okay. So, acids and bases, right? That's really what we're talking about with pH can do this. And we could talk about this more, if anybody wants to stop by for class in the open office hours. And then obviously temperature, really it's heat, right? So once you heat a protein past its critical temperature, it will change shape. Cold doesn't really do it so much, okay? It's really the heat that does it. So by vibrating that molecule, you can actually literally vibrate it out of its original configuration and denature it. Okay, so as I said before, proteins can be altered, I guess, by natural things that happen in the body. Now, I know we haven't talked about this yet, but you guys should know that, proteins that are made incorrectly in the human body can have major consequences. Here in this picture, sorry, I already had arrows up here, but here in this picture, you see a normal red blood cell. Kind of looks like I don't know, a cream donut with, that's kind of red, right? It's a little bit pushed in the middle. This is a normal red blood cell. Okay, it's round, so it slips through blood vessels without getting stuck, and it's designed to carry oxygen. This here is somebody that does not produce the proper amount of, or sorry, the proper sequence of amino acids when they build this protein, and therefore the protein is faulty. Okay, so we call this sickled a red blood cell, and this is what results from sickle cell anemia or sickle cell disease, as it says up top. These proteins were not made right to make up this red blood cell, and therefore it's this crescent shape, right? Now, the problem with a crescent shape is the very, very tiniest blood vessels in your body cannot transport this shaped cell very efficiently. So what that's going to lead to is basically it's going to clog up, the blood vessel, and that's going to lead to clotting, right? Clotting, not good, especially in blood vessels, simply because it's going to deny oxygen to cells that need it, right? So, in other words, if you have a clot in your heart, that leads to a heart attack. If you have a clot in your brain, that leads to a stroke. If you have a clot in your lung, that leads to a pulmonary embolism, which is also fatal, right? It can be fatal. Deep vein thrombosis. If you know any about the medical jargon, clots are really bad in the body, right? So people with sickle cell disease are constantly under the threat of developing a clot. And the reason for that is this up here is normal hemoglobin, right? The numbers that you see here are normal, the normal sequence of amino acids that make up the hemoglobin protein, okay? Look at number 6. Okay, number 6 has a GLU in it, which stands for glutamic acid. You don't need to know that, but in the people that produce, faulty hemoglobin, in other words, have sickle cell disease, see what happened here? They replace just one amino acid, right? Everything else is the same, but just one amino acid was, substituted. and that's called valine. And what happens there then is the entire protein shape, which is supposed to look like this one, now looks like this. That screws up how the cell transports protein and transports oxygen and also its shape. So therefore, people with sickle cell anemia have really awful problems getting enough oxygen to their tissues. Okay, so I think what I'm going to do is I'm going to stop here and we'll have one final video on DNA and RNA, nucleic acids. We're just going to talk about the molecule and what it looks like and its function, but some of the more detailed stuff that you may have learned in high school, which namely transcription and translation, we're going to save that, the details of that anyway for a later presentation. All right, so one more to go.